Hello? Sorry, this way. Sorry, yeah, someone was like calling me from outside. Hello, can you hear me? Fine. Hello, can you hear me? Okay, okay. Right. Uh, so, well, how was the exam? First of all, it's okay, it's okay. You can just text me about whatever you need, okay? Yes, first of all, how was the exam? I think it was yesterday, right? Or the day before yesterday? No, it was yesterday because we thought on. Uh-huh, uh-huh. Okay, is it MCQs or short answer questions? Like the questions where you had problems with, right? Is it multiple? Oh, okay, right. Yeah, of course. I think so, right? I think question practice is something we need to focus on, right? So, well, um, I guess because right we are like going coming up in like the middle of the course, right? We're teaching from the middle of the course. Uh, what do you would like to do, right? Because last class we're talking about tumors, right? Tumors and all those stuff, cancer and all those things, right? So would you like me to continue with protein transcription because that's like a very difficult topic, right? Transcription, translation, etc., etc. Or would you like me to focus on question practice, like how do you answer questions, MCQs and stuff? Translation. Yeah, of course, right? So, okay, I'm gonna open the whiteboard, and I will send the PDF file to your email, right? After every class. Yeah, sorry, last class I was setting up the Zoom meeting and stuff, and it took some time. But I think, yeah, the Zoom is like working as it is supposed to now, so I can send the PDF files right after the class. So, translation, protein synthesis. Give me a second. Which chapter is it? Here it is. I'm pretty sure you know the structure of the DNA. So we're going to go through that very quickly, and then we're going to talk about replication and protein synthesis. Okay. So you know what? Let me just focus on this idea. We have DNA replication, DNA structure, protein synthesis, and then obviously we have yeah, of course, it's fine. Translation. Let me know if you're back. Okay. Because yeah, I will have to work on your. Hello? Uh, So I am like, as you can see on the whiteboard, this is the basic structure of the DNA, and I'm like breaking it down, right? So a lot of the biological molecules that you're gonna study, including DNA, you can think of them as monomers joining together to form a polymer. And the easiest example is think about bricks, right? Climbing up to make up a building. Right? So you have one brick, another brick, a third break, fourth break, breaks. You join them together and you get a protein. That's it. This is how, right? The molecules are built. So whenever you study any molecule, it's proteins, right? Fats, DNA. Just think about single bricks, right? Single, like Lego bricks, right? Single components, monomers joined together to form a complex, large structure. Okay, so the nucleotide is the word we use. Okay, nucleotide is the word we use to talk about a single monomer, a single brick, basic unit of DNA, and it contains a five carbon sugar, phosphate group, and nitrogenous base. So in the exam, you will be asked to identify, right? Usually asked to identify the nucleotide. So for example, this makes up 1 unit of DNA. Okay, this makes up one nucleotide or 1 unit of DNA, and this makes up another nucleotide of another unit of DNA. So in the diagram, you can see here, right? You have two nucleotides joined to each other. And this is the structure of a single DNA strand. That's the structure of a single DNA strand. Do you have any questions? Okay. Um, what features do we have, right, of the DNA molecule? The DNA molecule is all, so we have this structure, and the third part is nitrogenous base. Nitrogenous base is like the flexible, changeable portion of the nucleotide, right? So the way it works is the phosphate group is always the same structure, PO4. 5 carbon sugar, right, is also the same structure, the same chemical, and then the nitrogenous base can be either adenine, thymine, cytosine, or guanine, right? That's how it works. Okay, that's how it works. So nitrogenous base part is always changeable, flexible. Okay, that's and we have like polynucleotide. Right? We use the word polynucleotide. Mhm. Many nucleotides joined together. Okay, that's how that's it. And they are joined together by actually phosphodiester bonds, which you don't really, oh yes, you do need to know, never mind. So phosphodiester bonds, so phosphodiester bonds, huh? Phosphodiester bonds join nucleotides to each other, right? So how does it work? It works by phosphate group, right? Joins with H2, right? 5 carbon sugar molecule on each side with an ester bond with one ester bond each. So there's a total of two ester bonds. You're supposed to know like the detailed chemistry, right? So if you look at this diagram, you can see that here we have a phosphate group. Here we have the right guanine. So this is an ester bond. This is an ester bond, and then the other one is also an ester bond. So we have two ester bonds, right? That's why it's called diester, phosphodiester, right? Phosphate and two ester bonds, right? Phosphodiester bonds linking the right two nucleotides. Any questions so far? Yeah, it's because just to make sure, right? You understand or whatever, right? If you yes, it's just type below. Yes, very good. Right. So we have that, and you do need to know the detailed structure of the right the molecule, and this is where carbon naming system is very important, right? Carbon naming system is like very important. So we say that this The structure of the DNA looks like this, right? Because the deoxyribose sugar has 5 carbons, right? It is very important for us to number the carbons so that we can accurately identify which carbon is forming which bonds. Okay, so this kind of carbon naming system is used whenever we have, right, uh, more than one carbon in an organism. So as you can see here, the naming works like this. I think it is 1, 2, 3, 4, 5, right? So that's how it works. So for one strand, right, for one strand on this side, you have start with the phosphate group, right? Phosphate group, 5' carbon, and this is actually, let me get this is six. Sorry, this is six. The naming goes like this. Yeah, right. So phosphate 5', 3', phosphate 5', 3', phosphate 5', 3'. Okay. So the structure, right? For one strand, the structure goes like this: phosphate group joined to 5' carbon with an ester bond. Don't forget an ester bond, right? And from that same sugar, the 3' carbon of that sugar is joined to another phosphate group, right? And then that's joined to 5' carbon, and that same sugar, 3' carbon, is joined to phosphate group, and so on and so on. This continues. Well, because DNA is a double helix structure, right? It's made up of two strands. The other strand, on the other hand, goes in the opposite direction, right? As you can see in this diagram, here, because if you look at the top, right? If you look at the top, here it goes like this: 3', 5', phosphate, 3', 5', phosphate, 3', 5' phosphate. Okay, so for the other strand, it goes like, right, 3', we start with 3', same sugar, 5' carbon, and then phosphate group, right, and that connects to 3' carbon and same sugar, 5' carbon, phosphate group, right, and then that connects to 3' carbon, same sugar, 5' carbon. So you can see that, right? They are like opposite. They're like kind of like opposite, right? The one strand is going 5 phosphate to 5' to 3'. The other one is going 3' to 5' to phosphate, 3', 5' phosphate, right? So here this starts with 3' carbon and ends with 5' carbons. And this one starts with 5' carbons and ends with 3' carbons, right? So because if you're looking starting from the same direction, right? 5, 3, 5, 3, 5, 3. Here is the opposite 3, 5, 3, 5, 3, 5. That's why we see that the two strands are anti-parallel. You have any questions so far? Okay. As for the nitrogenous bases are right divided into purines. Purines means they are they are made up of two nitrogen rings, right? And they contain adenine and guanine. Pyrimidines contains only one nitrogen ring, and it contains cytosine and thymine. And as you already know, purines always pair with pyrimidines, right? Adenine forms, uh, I think adenine is like one hydrogen, two hydrogen bonds with thymine. So you're supposed to remember these small little details, right? Guanine forms three hydrogen bonds with cytosine. You can see that in the picture here, right? So you have adenine thymine, two hydrogen bonds, and cytosine guanine, three hydrogen. You have any questions? Okay. This is called the complementary base pairing rule. Okay, this is called the complementary base pairing rule. Oh, sorry. I think my mouse is like auto-clicking. Okay. So we have that. And then you just need to know the general structure of the DNA, general features of the DNA, right? Double helix, double helix structure, a complete 10 turn of the double helix, right? Takes place every 10 base pairs. For every 10 base pairs, you have a complete turn, right? 10 base pairs, a complete turn, 10 base pairs, a complete turn, right? And each chain is a right-handed helix, right-handed, and we always have two nucleotides, right? Right. Two polynucleotides, right? And then chains are antiparallel, blah, and chains are attracted to each other by hydrogen bonding between the nitrogenous display. And because of that, because of the fact that, right, purines contain two nitrogen rings and pyrimidines contain one nitrogen ring, because of this, we say that there's always the equal distance, right? Equal distance. So the distance between two platforms is always, right, constant and the string rings one. Always. Because, look at this, sugar, thymine, right? Thymine is a pyrimidine. Do not forget this is one nitrogen ring, and this one is two nitrogen ring, and then we get the sugar. So that's three nitrogen rings. Here we have cytosine, two nitrogen ring, guanine. Oh, sorry, cytosine is a pyrimidine. My bad. Right? One nitrogen ring. Here two nitrogen ring, and combined is three nitrogen ring. You see that? Does it make sense? Right? So there's like equal distance, and then every 10 base pairs, you have like one turn. If you know that knowing that is very important because that will help us understand how the DNA replication and also transcription works. Okay, so you need to know the key enzymes that are responsible for replication, which in your book, let me check the syllabus just to be safe. Polymerase ligase. Okay, just leave the polymerase ligase, and then leave the blah blah blah. Okay, right. Uh, um, okay. So just know that various enzymes are involved in DNA replication, but you only need to know DNA polymerase and DNA ligase. Okay? You don't need to know other things. But the first thing you have to understand is that DNA exists as a double helix structure, right? Where the two strands are connected to each other, the two strands are connected to each other with hydrogen bonding, right? They are connected to each other with hydrogen bonding, right? Which is why you need to first, what we call, first, right? The first step in DNA replication is you need to unwind and coil the DNA, right? So to unwind the two strands of DNA, right? By breaking the hydrogen bonds that hold the two strands together. So in this context, right, we're gonna break apart this, this, this. Right? You have to break all the hydrogen bonds. And there's going to be an enzyme that is responsible for that, but you won't learn it here, right? And DNA polymerase follows the unwinding process and, right, synthesize nucleotides, oh sorry, joins nucleotides, so let's say joins nucleotides to form a new strand complementary to each strand, origin strand. And this process is called semi-conservative replication, but there's a lot of issues with that. Right? First, let's look at a picture, right? Let's look at a diagram, a picture, blah blah blah, and we can discuss that. Right? First, I'm going to give you the DNA code. Give me a second. I'm going to use this as an example. This is the DNA. Right? This is the DNA. Uh, and this is the complement. Okay, so this is the DNA strand, two strands. This idea applies for transcription too. Okay, do not forget this idea applies for transcription too. So kind of like we can basically cover both ideas at the same time. Okay, kind of, not really, but yeah, this is the end strand, right? What we want to do is we want to break it into. We're going to separate the hydrogen bonds. So we're going to separate the hydrogen bond into the two strands. So now it's separated. Okay, but the way it works is that part is a little bit confusing. If this goes from 5 prime to 3 prime, this goes from 3 prime to 5 prime. Plus the DNA is anti-pearl. Okay? So first you unzip, right? First you unzip, and then so this is 5 prime to 3 prime, and this is 3 prime to 5 prime. But the way it works is that polymerase can only copy in the 5 prime to 3 prime direction. Polymerase, right, can only copy in the 5 prime to 3 prime direction. Why? It has to do with the chemistry, the biochemistry, which is beyond our level. Okay, just the way chemistry works is it can only copy in the 5 to 3 direction. So for this one, it's very simple because the new strand, right? The new strand is going to be C A T C A T, etc., 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 etc. And here is 5 prime to 3 prime. So all polymerase needs to do is that they just need to join them together, right? So there's going to be a cytosine nucleotide. There's going to be like a random cytosine nucleotide, right? In the surrounding, because we have like so many nucleotides that are already exist, right? That already exist in the nucleus, right? They are already there. Do not forget, the nucleotides already exist. How do we get these nucleotides? How do they appear to begin with? Because we eat, right? We eat food, we get proteins, we get phosphates, we eat meat, we eat, right? Noodles, all the food we eat, the nutrients, the resources are used to make nucleotides. So the raw nucleotides already exist in the nucleus. They are already there. But what's going to happen is that once we, right, separate the strands, when we split them apart, when we split the strands apart, these nucleotides are going to fill in the gaps, right? So here's guanine, right? Here's guanine. So a random cytosine will come and attach to guanine, right? It will come to attach to guanine by hydrogen bonds. Oh, here's thymine, right? Here's thymine. A random adenine will come and attach to the thymine. Oh, here's, right? Thymine, it's going to attach, and attach. So all of these are held together by hydrogen bonds. Okay, hydrogen bonds. Just random attachment via hydrogen bonds. Okay, and the job of polymerase is to link the nucleotides together because each one is attaching separately, right? For each one is a separate attachment, right? Here's one hydrogen bond attachment, another hydrogen bond attachment, another hydrogen bond attachment, another hydrogen bond attachment. All of them are attaching separately, right? It's like the fingers, right? They're like all separate, right? What we want to do is we want to join them all so that now they form one continuous strand. Are you a little bit confused? Do you get it? Okay. So each nucleotide is attaching separately, and then we join them also, they form one continuous strand. By the way, this is also how transcription works, right? But except for transcription, we use mRNA instead of DNA. Okay. So for this side, we can do that. Polymers can do that because we just need to go from 5' to 3'. Okay, very straightforward. But for this side, polymerase faces a problem. Because polymerase can only copy from the 5' to 3' direction, this means for this, right, the strand, they have to copy this way. They have to copy this way. Okay. So for this one, polymerase, so the bond is going to be like this, right? G attached, hydrogen bond, P attached, hydrogen bond, C attached, hydrogen bond, and so on. And polymerase have to attach them this way. Okay. But the problem is that DNA strand is very long and you are unzipping. Basically, it's like a zip, right? You're unzipping the helicase is unzipping the DNA strands, right? And the unzipping process goes in one direction only, right? Here you can see with my foam pad, right? So you have like zip, then you have like unzip, right? It goes in one direction only, right? So for this side, polymerase is like copying in one direction, 5' to 3'. But for this one, because of the way the unzipping process works, right? You start from this point, you start the process copying from this point, but unzipping continues this way. You start your right copying here, but unzipping continues this way. Okay? So that creates a problem because your starting point is here, but there's more DNA code on this side. Which means polymerase have to choose another starting point again and copy again. Okay, and then there's more DNA and being acid, so they have to choose another starting point again and copy again, right? And that creates what we call fragments. That creates what we call fragments, and these fragments, right, because of the way the copying process works, there's like fragments. You can see that in the picture here, right? For this one, for the first strand, you just need to go right 5' to 3', just continuous right copying. But for this one, you choose the starting point. Here you have another fragment. Here you have fragment. Here you have one fragment. Okay, so you have all these Okazaki fragments, right? You have to copy it in batches, fragments, right? And that means you have to connect the Okazaki fragments, right? And the job of the connecting job is done by DNA like this. Right? So we have two types of strands. We have the lagging strand and leading strand. Right? Leading strand is where the parent strand that runs in the country direction. Right? The parent runs in the different direction. Right? And it's copied, right, by DNA polymerase in the 5'2" replied. Oh, it's just a name. Okazaki means just a name, right? It's like because the scientist who discovered this idea, the scientist who discovered that, right? They copy in batches. His name is Okazaki. He's a Japanese scientist. That's why we call his fragments Okazaki fragments, right? So at this point in the assembly process, you go this way, one fragment, another fragment, another fragment. So the scientist discovered that, right? For this side, you have fragments, and you have to join the fragments together. And the scientist's name is Ogasaki, so we call these fragments Ogasaki fragments. Okay? And for the next strand, which is what we are looking at, right, the thing B, right, is the parent strand runs in the 5' to 3' direction, right? Which causes polymerase to copy the, right? Make a copy in fragment, right? Which have to be joined together by likeness. A little bit confusing, right? That's how. Any questions? No. Now, just to because okay, we will do a formal question by this later, right, and stuff. But just to help you understand how these questions can appear, right? They might ask because there's like very pictures, there are a lot of pictures, there are a lot of right diagrams and stuff. They might expect you to know the 5' 3' this direction thingy, right? 5' 3' they want you to know the directions. They expect you to know the directions, right? So that you can answer questions about which way that the polymerase is going to copy and which way the polymerase is not going to copy. So, for example, uh, let me get an example. Okay, give me a second. Yeah, this question is like very common. Usually, they will expect you to do further thinking, right? They will expect you to do further thinking from what we have covered. So it's never the right way. The questions are never the right. So if you see something like this, you know that at the 9 is trying to timing and 1 9 is trying to cycling, right? Because at the 9 timing always goes like this. If you see a question like this, if 20% of the nitrogenous phases are at the 9, let's say this is 24, right? This is 24. That means there should also be 24. That means the remainder, 1 9 plus cytosine is going to be 100 minus 24 minus 24, right? So cytosine will be half that amount, 100 minus 24 minus 24 per 2. That's how they're going to ask the questions, right? They expect you to understand the process and they're going to ask further thinking questions based on what you have learned. You have any questions? No? Okay. Yeah, so it's the variable of mass. So let me do the math. Yeah, they should be, yeah, never mind. Yeah, it's B. Correct. Okay. Yeah, that's it. Right. And we're gonna see some examples later, right? But this is how the replication process works. Any questions? Anything so far? Okay. Now let's look at what we call semi-conservative replication. So the thing that we just saw, right? The thing that we just studied just now is called semi-conservative replication process. It means, right? DNA molecule is copied. Oh, sorry, it's gonna put it this way. The new DNA molecule synthesized contains one strand, one original strand, and one newly synthesized strand. Okay, this is the replication process. Okay, it's a replication process where the new DNA molecule synthesized contains one original strand and one newly synthesized strand. Okay, we already saw that in this picture, right? This is the original, right? This is the newly synthesized. Original, newly synthesized, Fragments, we have to join them together, right? So the new molecule is always made up of one original, one new. That's the semi-conservative replication process. But, okay, here's a big but. They will expect you to know the experiment. Mendelssohn, it's not in the textbook, which I find very surprising because they asked that in the exam questions a lot. Okay, they asked that in the exam questions a lot. So because I'm checking the past papers now, and they really expect ask a lot of questions related to that. Okay, so we're gonna look at Masterson and Stir experiment. How do we know, right? So this is what I've experienced, like what how it actually works. But how do we know? How do we know that, right? It is semi-conservative. So these guys, Masterson and Stull, they did the experiments. What did they do? Well, first they grew bacteria, okay, in a nitrogen 15 environment, which means whenever the bacteria divides, multiply, right? The DNA synthesized used nitrogen 15 for its components, right? So remember, nitrogen is present in nitrogenous bases. At the night time means hydrogen, quinine, right? They all contain nitrogen, right? Purines have two nitrogen means quinine has, primides have one nitrogen, right? So what we are saying here is that if you put the bacteria in a nitrogen 50 environment, so whenever it divides, right? All at the nine, all nitrogen, right? All nitrogen in adenine, thymine, guanine, cytosine, etc. will contain nitrogen 15. Right? Do you have any questions so far? No. Okay. And then what they're going to do is that they're going to take the bacteria from the nitrogen 15 environment, and we're going to put them in the nitrogen, I forgot it's 14 environment. Now here's the issue. Now the bacteria will have to use nitrogen 14 to make DNA, nitrogenous bases, since nitrogen 15 is not required. Okay, so they're going to ask a lot of questions. This is the experiment, right? They're going to ask a lot of questions related to that. And if you know the basic idea, you can make predictions. Okay, so there are three theories. In the beginning, there are three theories: conservative replication process, semi-conservative replication process. Let's ignore that for now. Conservative replication process says that, right, after DNA is replicated, there will be one O molecule and one new molecule. Okay, there will be one O molecule and one new molecule. Semi-process conservative replication process states that, right, just look at that, if replicated, there will be one, the new strand, right? Every new strand, every new molecule contains one original strand and one new strand. Okay, so which one is the correct version? That should be interesting. Okay, let's put it this way. I'm going to use color, right? I'm going to use color for this. If conservative is true, right, the original DNA will contain nitrogen 15. Okay, color coding is red contains nitrogen 15. Okay, the original DNA contains nitrogen 15. Right? But do not forget, we transfer these bacteria into a nitrogen 15 environment. We transfer these bacteria into the nitrogen, sorry, not 15, sorry, 14 environment, 14 environment. Right? So if conservative is true, we should observe two original strands, okay, and two new strands, like this. Let's change the color. So, do you have any questions for this one so far? Hmm. On the other hand, if semi-conservative replication works, then it will be more like this. You get that? Okay, right. So I want you to be able to answer questions, right? Because that's like, yeah, a little bit confusing. So we can look at this. If someone is, give me a second. All right, right. What was in the last test? Yeah. Did you get it? No. Okay, I know. It's hard, right? It's like extra thinking. Uh, yeah. So let's look at this, right? Here, here's the question. So I think it's not just about memorizing the facts, right? You also have to do um the thinking, right? That's why I think in our revisions, when I'm like reviewing all of this with you, we will focus on question practice as much as we can, right? But uh, yeah, so here you can see that, right? So based on everything I've explained, based on everything that I've explained, can you answer this question? What do you think the answer is? Oh, you're gonna see it's 75 and 50. Let me type it. But you have to understand that, right? You're talking about the five once and the five second time. You think like the five once and the five second. Well, you already know that sum equals of this is the correct one, right? No. Okay, but let me explain. I know it's a little bit confusing. Do not forget, it's the second division we're looking at, right? But let's start with the first division. Yeah, let's start with the first division, right? If semi-conservative is correct, right? If semi-conservative is correct, all of them, all of them should be like this. Do you understand that? All of them should be like this. So after the first division, it should be 100% hybrid. 100% hybrid. If semi-conservative is correct. Are you confused? Okay. But they are not asking about the first generation, they are asking about the second generation. Okay, they ask about the second generation. So let's talk about what happens after this. So remember, we have like in the next division, right? The next division, replication. Now we're going to separate this. Remember, this is the original. Do not forget. For the second generation, this is all the original, right? This is all the original. All of these are the original. So you separate the originals, you separate the original strands, you separate the original strands, right? You see that? And then you're gonna put a new strand here, you're gonna put a new strand here, you're gonna put a new strand here, you're gonna put a new strand here. That's how semi-conservative replication works, right? Separate the original strands and put in the new strands. Okay? Separate the original strands and put in the new strands. I confused. Do you have any questions so far? Here's my question. The bacteria is it in the nitrogen 14 environment or nitrogen 15 environment? The bacteria, right? Is it in the nitrogen 14 environment or nitrogen 15 environment? According to the question, right? We transfer them to the nitrogen 14 environment. Uh, look, uh, look, I did. Ah, okay, okay. Because I saw you talking to someone, so I thought you're like, yeah. So it's okay, right? Let me continue. So I was telling you that, right? The bacteria is in the nitrogen 14 envelope, right? This is straight after the first generation. This is straight after the first division. And for the next division, okay. Oh, sorry. For the next division, what raw material is the bacteria going to use for the next division? Is the bacteria going to use nitrogen 15 as a raw material, or is it going to use nitrogen 14 as a raw material? 1,4 or 1,5? Very good. So we separate the original strands. We always have to separate the original strands, right? And we're going to make a new strand, new strand. So the new strand that is going to be here is going to be blue, the blue, right? What was it? This is very dark blue. Let me change the colors. Is this blue? Oh no, not this blue. No, never mind. I'll just copy this. Okay. So the new strand that's going to be inserted. That's going to be inserted by synthesized. It's going to be nitrogen 14, nitrogen 14 strand, nitrogen 14 strand, and nitrogen 14 strand. You see that? Hmm? This is straight after the first division. This is straight after the second division. Okay. As you can see here, after the second division, we have one, two, three, four, five, six. Six nitrogen 14 strands. And we have the original from the original, right? From the very, very original, nitrogen 15 environment, 2, 15, and 15 strands. Okay? This is from the very original, in the very beginning, right? The bacteria grew up in the nitrogen 15 environment. And then we transfer them, we do one division, insert 14, insert 14, and then we have to separate it again and insert 14, insert 14, insert 14, insert 14. So we have 1, 2, 3, 4, 5, 6, 2, 3, 4, 5, 6, 6 for nitrogen 14 and 2 nitrogen 15. Get it? And that's why the ratio is going to be 6 by 8, right? And 2 by 8, which is, so what's the answer? What do you think the answer is now? Let me go check the answer. Oh, that should be C. No, it's not C. Wait a second. Mhm. Yeah, it's D. It's D. Yeah. Right? 2, 3, 4, 5, 6. Start 8. 2 by 8. 2, 3, 4. 1, 2. Yeah. It's got a D. The answer one is D. Because this is asking about, I know the vocabulary is like, yeah, vocabulary is very confusing. I was also very confused. This is asking about the strand. It's all about like reading, right? It's very hard. It's like IELTS. This is asking about the strands and this is asking about the molecules. They ask me two separate questions, right? So if you calculate the strands, if you calculate the strands, right? Strands is one, two, three, four, five, six, seven, eight. Eight strands, right? And of the eight strands, six are light, right? So that's six by eight. But this question is asking about the molecule. If you ask about the molecule, you have to look at this. This is one molecule, another molecule, another molecule, another molecule. Two strands combined is one molecule. Two strands combined is one molecule. Do you understand? Two strands combined is one molecule. So how many molecules do we have? We have four molecules. And out of four molecules, two are hybrid. So that's two by four. Yeah, I was also confused because I wasn't reading carefully, right? See, it's the tiny little details that can make you lose marks, right? First of all, you have to understand the concept, right? The concept is very important. And another thing is the wording, right? Strand, molecule. If you misunderstand that, you're gonna be like, okay, this is right. The way I thought too, right? I was also very confused because I was like, oh, it's just by eight is right. Light is just by strand is just by eight, right? Uh, Heavy strand, sorry, not hybrid, like it's hybrid, sorry, hybrid. Oh yeah, like this. Hybrid strand, hybrid is heavy is Dubai. That's what I was thinking. But they're not asking about the strand, they're asking about the molecule on the right hand side. So just read the question very carefully and highlight all these keywords. Right? You can make you feel well, very confused. Even if you already understand everything. But this is everything you need to know about some because of different repetition. I think you also know how to do these kind of questions now, right? And just to help you understand, how do we find out this hybrid heavy, right? How hybrid light, blah, blah, blah. We use a centrifuge, right? We use a centrifuge, right? So I hope you already know what a centrifuge is, right? We just put it in like a blender, kind of like a blender like thingy. It's going to rotate and the heavier things will settle at the bottom, right? So let's try another question like this, and then we can move on to transcription. Oh, scroll up. Let me scroll down. Right, that's very like this one. Can you see now? No? Ah, okay. It's a D. Okay. 23, right? No. Let me see carefully. Okay. Um, Remember, after two divisions, it should be like this. It should be like this, right? You might want to. You understand? After the second division, it should be like this. After the first division, it should be like this. Did you understand? And this is why you want to read the text very carefully. What do they say, right? What does the question say? Yeah, I get that, right? So that's why it's like you need to know the concept and you have to understand what they are saying. They said heavier molecules are close to bottom, right? So you can see that, right? M is heavy. Yeah, M is heavy. And that means, right? Light is, oh, that means light is up, right? Because you see that heavy is down, so light is up. Did you understand that? So light molecules should be at either K or L. Okay, there are only three possible types. There are only three possible versions. Either you are completely heavy and 15, or you are completely light and 14, or you are hybrid, which is N14 plus N15. Does it make sense? And since there are only three bands, there are only three bands, one, two, three, and M is the heaviest. M is the heaviest, right? So it's first you have to start with the thinking, right? First you have to understand where they should be, right? So since M, the heaviest, is at the bottom, what should L be? Should L be completely light or hybrid, right? If you have something here, is it this or is it this? Did you answer my question? Yes, very good, right? So you know that L should be hybrid, and then K should be very good, right? K should be light. If you understand that, you just go up and match it with what we know. After the first division, we only have hybrid. Do you see that? After the first division, we only have hybrid. So KLM. Where would it be? After the first division, KLM. After the first division, should it be at K or O or M? Yes, O, right? Hybrid from here. So that means after the first division, it should be hybrid or O. Exactly. And after the second division, now you can see our picture, right? Sketch it. In an exam, sketch it because it gets very confusing. I also get very confused. For me, right, 15, 14, that's very confusing. So you want to do something like this in the exam. Okay? So after the second division, what do we have? Remember, there are only three possible versions. Hybrid, or completely light, or completely heavy. Right? So there are only three possible versions. So here, if you look at this, what do you see? Molecules, right? We look at the molecules. You see two completely lights, two hybrids. Right? After the second emission, you have two completely lights and two hybrids. So in terms of bands, where would you find them? Two lights, two heavies. So M is completely heavy. Right? M is completely heavy. Do remember we have two completely lights and two hybrids. Oh, yeah, sorry, I meant hybrid. Ah, my brain. I meant hybrid, not heavy. Sorry, that's my fault. I made you confused. Right? So, like this, question like, where will we find like KLM? Where will we find like KLM? Completely like will be found at K, and two hybrids will be found at. Okay? So the answer is no, no, the after first division is all hybrid. After first division is all hybrid. Yeah, did you get it? This is after first division, right? This is after second division. Are you getting the thinking process now, or is this a little bit confusing? You get it right? So most questions you'll see in the exam is gonna be variations of this. It's the same idea. It's the same idea, but they're gonna test it in different ways, right? Calculate the percentages, right? Tell me where you will find it on the subject essays. Of course you can. Just I send it now. I send it still send it right.
I have an exam question that I will send you, but I will try to find them.
Ah, okay, okay. So you can maybe send it to me later, right?
Yeah, yeah.
Okay. All
Thank
right.
you.
Yes, yes. So I'll continue with transcription. I think that's enough. I hope you get an idea of right how this works now. The rest we just want to, uh, well, I guess like practice a lot of questions like that, right? Do not forget that you should first be to answer this, right? To get a correct answer, you need to do two things. First of all, you need to understand what will happen after the first division, what will happen after the second division, third division, and so on. You want to sketch something like this, right? Have it on your like notes, right? Sketch it in the exam so that you really know what's going on. But the second part, that's the more difficult one, is you have to read the question. Because sometimes, even if you understand, the wording can be confusing, right? Here I was stuck because I was like, I didn't see it. I was like, okay, here's strands, right? It's confusing. Here they are asking about molecules and strands, right? And here they're talking about centrifuge positions, right? KLM. So read the question very carefully, try to get it, but also try to right understand what it should be, right? You have to link the ideas like that. So now we're going to talk about transcription. Sorry, before we talk about transcription, we're going to talk about the genetic code, right? Genetic code is very important. In the exam, they might give you the entire genetic code. Okay, they might give you the entire genetic code, or they might give you like some short phrases, right? Short phrases like this. Um, yes, here's a possible question. Very confusing, right? Uh, so they're gonna give you something like this, right? But for now, we're gonna walk with the full genetic code. Okay, I'm gonna show you how to walk with the full genetic code, and then we can look at questions like this to know how to answer them. So wait, I'm copy pasting the stuff. Okay, give me a second. This is for. Do you see the images? I think if you're checking on your phone, they're like not like small. But can you see all three images? DNA code, RNA code, and the DNA strand. Yeah. Okay. So let's focus on this. So basically, the genetic code, as you can see in the picture, works like letters, right? You combine letters, they can create information, right? Like CAD cat, UD dog, RD rat, right? UD dog or UD god, right? The way you arrange the letters change the meaning of the words. The way you arrange the letters change the meaning of the words. So the code, genetic code is just like this, right? You look at the right nitrogenous species. Here we have 1,9 and a 9, 1,9. Okay, these three together, right? Responsible for a certain kind of amino acid. Cytosine, 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 that's responsible for another kind of amino acid. Thymine, cytosine, and adenine, that's responsible for another kind of amino acid. Blah, 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 blah. Right? So the genetic code, the DNA code, helps you, codes, right, helps you to determine what kind of amino acids you should be producing. And we're going to look at the features of the genetic code. What are the features of the genetic code? They are, number one, is the triplet code. All genetic code is a triplet code, right? That means three bases determine code for one amino acid, which means the order in which three bases appear determine which amino acid will be produced. And the code is universal. That means it is interpreted more or less the same way by every living organism, no matter the species. Technically, there are exceptions. It is, but we don't know about them. Okay. Another thing is the genetic code has uh, start signals and stop signals basis. Some codons, right, have start signals or stop signals. What it means? What does it mean? The codon doesn't code for an amino acid. Instead, the codon tells, right, the cellular machinery, right, the machinery to start transcription or to stop transcription. Right? It doesn't actually code. And then the genetic code is redundant or degenerate. We're going to get that later. Okay. Uh, but we're going to talk about this idea in a while, right? But for now, do you have any questions here? No. Okay. What does redundant or degenerate means? As you can see here, redundant or degenerate means more than two codons can code for the same amino acid. More than two codons can code for the same amino acid. Right? Here, timing, timing, timing. If you see three timings in a row, that's a signal that you have to do phenylalanine. You have to make phenylalanine. Okay? You have timing, timing, timing. That means you have to make phenylalanine. But also, if you see timing, timing, cytosine, timing, timing, cytosine, that also calls for phenylalanine. So this and this is the same. This and this is the same. You get it? Same meaning, different codes. And here we have timing, cytosine, timing, that calls for serine. Timing, cytosine, cytosine, that also calls for serine. Timing, cytosine, adenine, calls for serine. Timing, cytosine, guanine, that calls for serine. Okay? All right. So knowing the genetic code is important. And let me just check the syllabus a little bit. Okay. Right. Uh, and as you can see, if you go look at the genetic code, you can see that some of them are stop codons, right? TGA means you stop the process. TAA, TAG means you stop the process. This is something you need to understand. ATG calls for methionine, but it's also a start codon. Okay? ATG calls for methionine, it's also the start codon. Okay? Which means the first amino acid in the chain is the is methionine because it's also the start codon. You start with methionine, that's the starting process. Always no exceptions. All right. What does universal means? Universal means this process works the same way in every living organism. So if I have TCT, TAT, TGP, TGA, that means I'm going to code for phenylalanine, join to tyrosine, join to cysteine, and then there's a restop codon. So restop. Sorry, let me start with butane. I start codon is butane, ATG, sorry, my bad. Okay. So this is going to be my minus it. Okay. So ATG calls for start, TGA tells you to stop. This is what I'm looking at. In the exam, they might give you that. Okay, so they can either give you a table like this, can either give you a table like this, or they can also give you information like this, right? You do not need to memorize the genetic code. That's not required, right? But you need to be able to work with it. You need to be able to look at the information. You get it? What if we have something like this? Now you do the coding for me, okay? So if I have DNA code is like this, can you tell me the amino acids we're going to get, the amino acid chain that we're going to get? Can you write it down? I mean, you don't need to write the full thing, right? You don't need to write me down. I just follow the table and then just write MET or whatever. Yeah, yeah. For example, okay, let me give you another example. If I write ATG, because that's always the start code, right? CTA, TGC, TAA, that means methionine, arginine, TGC is cysteine, right? Now you give me something like this, because that's going to be the possible exam question, right? Now you give me something like this for this code, right? What is this going to give me? Okay, okay, very good. Methionine, right? The DT is uh, lysine, and then proline, CCC is proline, And then like C, oh, you don't need to write the stop. Stop means to stop, right? Stop doesn't mean this and the minus. Stop just means stop. So be very careful in the exam, right? So you don't need to write stop because it just means you stop copying the copying process. So this is the answer. Okay? If it is like a, if you have to write it down in the exam, this is the answer. I the answer now? Are you confused? Right? Okay. And universal means okay, so to copy your answer, universal means bacteria, E. coli will also produce this chain. Humans will produce this chain, right? Cats will produce this chain. That's what we mean when we say the DNA code is universal, right? Humans, lions, chickens, bacteria, all of them will produce the same chain. It works the same way in all of these organisms. That's how the genetic code works. Any questions? Okay. So with that knowledge, let's move on to the idea of we're going to talk about the transcription translation process in detail later because we're going to first get the coding process, right? If we get it, we need that to understand translation. Okay? But the way it works is that, I mean, you already have some idea because transcription is very similar to DNA replication, right? Very similar to DNA replication, is that you're making an mRNA copy. You're just making an mRNA copy. And the way it works is it's going to be DNA to mRNA to protein, but also tRNA is also involved in the process. It's also in the process. Okay, so the job of tRNA is to bring amino acids, right, that matches the mRNA code. So which means you have to go through multiple levels of thinking. You have to go through multiple levels of thinking. Okay, and it works like this. First, we're going to start with the DNA. Let's use the DNA code that I have before. Oh, what's my example? Oh, here. Let's use this code, right, as a template. Okay, we're going to use this. Right, this is a random made up thingy, right? So for now, let's ignore that, right? We don't really think of the logic. This is a random made up. First, what we do is we have to choose one strand as the template. And it really depends on the gene. Really depends on the gene. For now, let's assume, okay, I'm gonna add a ending code just to be. For now, let's assume I'm gonna choose this one as the template string. That means the bottom one is called a non-template string. Some you might see like different words, right? In the textbook, in the thingies, there's gonna be like different words. Sometimes you might see the word non-template strand, non-transcribed strand, right? Yeah. So in the book, they see the word non-transcribed strand, right? Non-template or transcribed. Yes, transcribed. So when we say the top is the template strand, what we are saying is that the mRNA produced is gonna be copied from this. mRNA is going to be copied from this. Okay? So it's going to be complementary. Don't forget this idea of complementary, right? So complement of C is G. Complement of A is U, uracil, because RNA doesn't have timing. RNA only has uracil, right? A, G, U, A, C, C, U, G. You see that? This is the mRNA copy. I confused so far. That's where people get confused. This is what people get confused. Even people who understand everything, they can get confused because it's a little bit confusing. As you can see, this is this is used as a template. This is used as a template, but the code on the mRNA matches the non-template strand. right? You can see that the only difference here is the thymine and uracil. Everything else is the same because alanine doesn't have uracil, right? So what is going to happen is that, yeah, that's the confusing part. Non-template strand is the code for amylin. Template strand complement amylinase, right? Complementary amylinase. And that amylinase actually code is reflecting the non-template strand. So that's the first thing you have to get. Any questions? Another thing is threonine. Threonine matches to the amylinic code and brings the associated amino acid. That means, right? And this all works in a complementary, right? It's always a complementary base pairing. So if this is the amylinine, the threonine that matches is going to be, it's called anticodon. This one is codon. So this is going to be the complement. This is going to be the complement. So C A U C A U G G A C. You see that? And threonine carries the amino acid. Threonine carries the amino acid. And the amino acid, if I follow the genetic code that we just looked at, not this one, this one. Well, we have an RNA code and we have a DNA code, right? But it's complementary, so it's the same. But the DNA is going to carry CAU, which is CAU is uh, what is C? Oh, histidine, right? And then GA is glycine. And then we have like an extra, right? We have an extra because remember this is a randomly generated thingy, right? So yeah, there's one extra. We ignore the extra for now. Okay. Do you have any questions so far? No, right? Okay. And this is they're gonna ask a lot of questions based on this coding process, right? Like I told you, they will either give you the entire thingy, right? Or they might just give you like they might give you the entire table. They might give you the entire table and make you do calculations based on that, right? Or they might just give you like a small, short uh phrase or whatever, and you have to answer questions based on that. So it really depends. So here's an example of question that you might see on the exam. Can you answer 22? Okay, this is the transcribed strand. Okay, this is the transcribed strand, which means it's the template strand. Transcribed is the template strand. No, wait, let me double check. No. First, we have to the transcribed strand, and then the complementary is the mRNA. There's a transcribed strand. You know what? Let's do this step by step, right? Transcribed strand, you're going to complementary, right? There's going to be a complementary mRNA, right? So what is the mRNA that you're going to get from this transcribed strand? First, let's find the mRNA code. What's the mRNA code? Can you tell me the mRNA code? Oh, no, 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 not that minus it. Okay, so here we have like CTG, right? What's the complement of that? What's the complementary function of that? Opposite of C is what was B? No, image is okay, right? Yeah. All right. So GCC, yeah, you keep thinking like that. GCC and the flip function is so the second one, I guess the first CCC. Yes. So it's the GCC, and then the next one. Can you continue? The next one is perfect. Yes, yes. Although I like the last one is wrong. GGC and GCC. Did you get it? Right? Because it's a transcript strand. It goes like this. After this one, I'll got this. And then just do the matching thingy, right? GCC calls for alanine, right? And then the CGG code for CGG, what's CGG? Ah, here, arginine. GGC code for GGC code for glycine, and GCC code for alanine again. I know, because of this word. Transcribed strand means from that you get the mRNA, and from that mRNA you get the protein, right? I think what you are trying to do is that you're trying to get the amino acid provide directly from this, right? I think that's what you're doing, trying to do. Okay, be very careful with the wording. If the question says, okay, if the question says non-transcribed strand, and understand that if this non-transcribed strand is the same as the mRNA code. Okay, transcribed strand is the complement of the transcribed strand is the mRNA. If you understand that, you can answer questions more easily. Okay. So if the question says it's non-transcribed strand, we can just say CTG. You can just look at that, right? That is going to be right. Arginine. GCC is alanine, right? We can just think like that, right? So if it's transcribed, we just treat this as like an mRNA. If it's non-transcribed, we have to first get the mRNA first, and then get the amino acid. You get it now? Right. That's why you understand the concepts, but also read the question very carefully. You see that? I think some of your mistakes have to do with not reading the question very carefully. Even if you understand, if you don't read it, if you don't get the keywords, you're going to make mistakes. Do you see that? Okay. So let's stop here for today. I will send you the yeah, I know you have to highlight it. You need more practice. Practice more with a lot of questions, right? And then you'll get used to it. But now I think you get the thinking now. All right, so we're gonna stop here for today. I will send you the PDF file, everything, right? So see you
Thank you.
next week.
Thank
Bye.
you. See ya.